Okay, we will be studying most of the experiments from the experimental biochemistry and biotechnology series 1. Today we are going to talk about the experiments of carbohydrates. We will be talking about the scheme of carbohydrate experiments, and we will also talk about the Molisch experiment today, and we will talk about the iodine test. We will talk about the Fehling test that is utilized to find if the sugars are reducing or not. And we will also talk about the Barfoed test that is utilized to identify if the monosaccharides are there in the sugar in the carbohydrate or not. Seliwanoff test we will be talking about. This is utilized to identify the keto group, and the pentoses are identified with the help of Biles test. And at the end we will be talking about the quantitation of glucose by the DNS reagent. So, what is the scheme of study for the carbohydrate analysis? If an unknown carbohydrate is given to us, we have to analyze under a certain scheme. First of all, we have to know whether it is carbohydrate or not. We can be given a solution of protein, we can be given a solution of carbohydrate, we can be given a solution of lipids. First of all, we have to know whether it is carbohydrate or not. The Molisch test is performed, that is only giving positive test for the carbohydrate. It gives only positive test for the carbohydrates. First of all, we will perform the Molisch test. If the carbohydrates are confirmed with its positive indication, then we will move on to see if these are polysaccharides or these are not polysaccharide. To identify the polysaccharides, iodine test is performed. Iodine gives deep blue color with the polysaccharides. If the deep blue color is there, the carbohydrate is polysaccharide. And if the deep blue color is not there, then it is not the polysaccharide. If it is not polysaccharide, we have to know whether it is reducing sugar or not reducing sugar. Like reducing sugar is glucose, maltose, and not reducing sugar is sucrose. To know whether it is reducing sugar or not, we have to perform the Benedict test. If the Benedict test is positive, it means it is reducing sugar. For example, it can be glucose, it can be maltose, it can be some other galactose. Okay, if it is not for positive test, it is reducing sugar. Then we perform the Barfoed test. When we perform the Barfoed test, it gives whether the carbohydrate is disaccharide or monosaccharide. If the carbohydrate is disaccharide or monosaccharide, Barfoed test give negative result for the disaccharides. It gives negative results for the disaccharides. Who? The Barfoed test. But for the monosaccharide, it gives the positive test. We will check all these experiments today. If the monosaccharide is there, whether the monosaccharide is aldose or ketose. Whether next test is Seliwanoff test. It is negative for aldoses, it is positive for ketoses. This way we know that whether it is ketose or whether it is aldose. Okay. One
Yes,
is
sir.
Ossazone test is there. Ossazone test gives the crystal in 10 to 12 minutes for lactoses, and it gives sunflower shaped crystals in 10 to 15 minutes for maltoses. We only perform this kind of test for 10 to 15 minutes only when the disaccharides is there. For the monosaccharides, Ossazone test is still performed, but it gives crystal too much early. Like for glucose, it gives crystal, needle-like crystals in within 5 minutes. But for other monosaccharide, which monosaccharide? The fructose. For the fructose, it gives crystal in 2 minutes. This is the scheme that we follow to identify what kind of carbohydrate it is. Let's see each experiment one by one. This is the first experiment. This is the Molisch test. This is utilized for the identification of carbohydrates. We can identify carbohydrates from the proteins, from the lipids by the use of Molisch test. Okay. What is the principle of this Molisch test? This is the equation that is showing the principle of the Molisch test. D-glucose in the presence of concentrated sulfuric acid is converted into the hydroxymethyl furfural, that upon its reaction with the alpha naphthol is converted into purple or violet color product. Glucose is colorless, is transparent solution, it is colorless, but when the Molisch reagent is added to it, the carbohydrate, the glucose gives the violet color. This is the confirmatory test for the carbohydrates. Okay, so what the materials are required? We need test tubes, test tube holders, sulfuric acid, stirrer, alpha naphthol, burner, and glucose. Okay, that take 2 ml, take 2 ml. So we have to read the protocols now. Take 2 ml of solution, take 2 ml of test solution with the help of pipette in a test tube. Take 2 ml solution in test tube. Then add 2 drops of Molisch reagent. 2 drops of Molisch reagent. It will be given to you in the laboratory, and mix it thoroughly. And mix it thoroughly. Then slowly add 1 ml of concentrated sulfuric acid. 1 ml of sulfuric acid, right? 1 ml sulfuric acid with the help of dropper, and do not shake. A reddish violet ring will be formed at the junction of two solutions when we add 1 ml of concentrated sulfuric acid. Violet ring will be formed. This violet ring is the confirmation that it is the carbohydrate. Okay, let me see one message. Some girl is sending. Yes, class start हो गई है. Please बता दो कोई बच्चा. Okay. So what are the results? Formation of deep violet ring at the junction of two layers shows the presence of carbohydrates. These are the results. When you are performing this experiment in the exam, always give heading of result and always write the result, because result have separate score. Okay, the reagents, Molisch reagents can be prepared by the use of 5% alpha naphthol in the 95% ethanol, and we also need other reagent, that is sulfuric acid, for this experiment. What is the significance of this experiment? This is the preliminary test for the identification of carbohydrates. It gives positive results for both free and the conjugated form of carbohydrates like the glycoproteins and glycolipids. So it is important to know that it gives positive results not only for the free carbohydrates but also for the conjugated carbohydrates. You already talked about the glycoproteins and the glycolipids. One exercise I always gives: take sucrose, glucose, and cooking oil in different test tubes and compare the reaction of each with the Molisch reagent. Compare the reaction of each with the Molisch reagent. So next experiment, if the carbohydrates are confirmed, we have to know whether these are polysaccharides or whether these are oligosaccharides. To confirm whether the carbohydrate is polysaccharide, we have to perform the iodine test. The principle of iodine test is that iodine is giving the deep dark blue complex with the polysaccharides. Iodine solution gives the dark blue complex with the iodine solution, the polysaccharides. These are the materials required to perform this experiment. This is the iodine solution is required, test tubes are required, burner is required, and starch is required. Starch is the polysaccharide that will be utilized to perform this experiment. Starch is the test solution in this experiment. Take 2 ml of same sample solution. Take 2 ml of sample solution means starch solution. Starch, sucrose, and glucose. You know, sucrose and glucose are not polysaccharides but starches. Starch is polysaccharide. Starch will give only the deep blue color. Rest, glucose and the sucrose will not. Add 1 to 2 drop of diluted HCl and mix it thoroughly. HCl actually facilitates the reaction of iodine with the polysaccharides. After the addition of HCl, add 1 to 2 drops of iodine solution and mix and observe the color. The deep blue color will be produced, which will disappear on heating. This is very fantastic. As a deep blue color upon the addition of iodine will appear. If you heat this deep blue color reaction, the deep blue color will disappear, and when you cool down the reaction, the deep blue color will reappear. It will reappear again. Okay, this is fantastic, very beautiful experiment. The color of the solution will be dark blue at low pH. Usually at low pH, its color is dark blue, and we added HCl for the dark blue for the low pH. Okay. Next is iodine test for the identification of polysaccharides. Okay, this is the same. Uh, Glucose shows the yellow color. Result is that glucose shows a yellow color. This was the actually glucose. This is the real experiment that I performed 1 or 2 years back in the laboratory. That glucose gives this kind of color, and the starch gives so much deep, dark blue color, almost looking like black. Okay, so you have to take this kind of picture when you are performing this experiment. So the reagent starch solution is required. 1% starch solution is utilized, and the recipe of the starch solution is given here. And the iodine solution is required, and the iodine solution recipe is given here. And you also need sometime iodine solution is made by this recipe, and sometime it's made by this recipe. Any can work. Significance. It has a significant role to follow the path of polysaccharide hydrolysis. If we are performing the hydrolysis of polysaccharides, we can know whether the hydrolysis of polysaccharide has been completed or not. If the hydrolysis of polysaccharide is completed, no blue color will appear. And if the hydrolysis of polysaccharide is not completed, then the deep blue color will be appeared. In this way, we can follow the hydrolysis of the polysaccharide by the use of iodine test. There is one exercise given. Compare the color of starch, maltose, and amino acid. You know, amino acid will not give this test. Amino acid will not give this test because amino acids are not the carbohydrates. These two can give some, only starch will give, and this will give maybe the yellow color, maltose. Okay, next is the Fehling test. Fehling test is performed to know whether the carbohydrate is reducing or not. Definitely, polysaccharide will not give this test. Polysaccharide will not give this test. Sucrose will not give this test because sucrose is also non-reducing sugar. And you know, glucose will give this test, maltose will give this test, and all the carbohydrate that has free anomeric carbon, that will give the Fehling test. The principle is that the cupric hydroxide Cu Cu plus 2 cupric hydroxide is converted into the cuprous oxide. This cupric oxide is of red color. This appearance of red color, that cupric is reduced by the carbohydrate, in this case is D-glucose, under the presence of low pH, in the presence of alkali. The red PPT are appeared. That is indication that the carbohydrate utilized in this experiment is reducing in nature. So this is the experiment. These are the reagents required that will be provided to you in the laboratory. Test tubes are required, beaker is required, and burner is required. First of all, take 0.5 ml of the sample solution. It can be the sucrose, it can be the glucose. Sucrose is non-reducing and glucose is reducing sugar. Take these sample solution in separate test tubes. Take 1 ml of each of the Fehling solution A and B in test tubes. Pour 2 ml of Fehling solution in each of the two samples. Add 1 ml each of Fehling solution A and B in both the test tubes. Keep the test tubes in boiling water. There is one test tube, one is containing the sucrose. Okay, the another test tube that is containing the glucose. Okay, and add both A and B here and both A and B here. What A and B? Fehling solution A and B. When we will boil the reaction, this reaction for 5 minutes, what will happen? The formation of reddish PPT indicates the presence of reducing sugar. Red PPT will be formed only in the glucose, and no red PPT will be formed for the sucrose. It means the glucose, the sample number 2, is containing the reducing sugar, that is giving you positive test for the Fehling experiment. So what kind of reagents are required? Results are there. Non-reducing sugar will give light violet color. Okay, this is the actually glucose. This is the sucrose. Non-reducing sugars will give this light blue color, and this kind of reddish, yellow to reddish color will be given by the reducing sugar. These are the real experiment that I performed, and we took the picture, and I'm showing this picture to you. And the reagents are required. The Fehling reagent A is required. That has the recipe of this one, and Fehling solution B is required. And significance of this experiment is that Fehling reagent is reactive towards the aldehyde groups containing reducing, but not to the ketosis sometime. Okay, reducing groups. So the exercise is that take 50 ml of each of the fructose, sucrose, maltose, and glucose and compare color change with Fehling solution. So until now, we confirmed whether it is carbohydrate or not. Carbohydrate. Then we confirm whether it is polysaccharide or not. Then we confirm whether it is reducing sugar or not. We are moving forward now. The next experiment is if the sugar is reducing or non-reducing, whether it is monosaccharide or not. Barfoed test is positive. Barfoed test is positive only for the monosaccharides. What is the principle? That D-glucoses in the presence of cupric oxide give the cuprous oxide, that is red color PPT. That is red color PPT. Okay, so we will see how to perform this test. This Barfoed reagent will be provided to you. Take 2 to 3 ml of Barfoed reagent and then add 3 to 4 ml of the 3 to 4 drops of the sample solution in the test tube. Then incubate the test tube in boiling water for 3 minutes. Boil the sample for 3 minutes. Deep blue color will appear. Deep blue color with the red PPT will be settled at the bottom. Blue color will be there when the precipitate will be of red color, and red color precipitate will be because of this cuprous oxide, will be because of cuprous oxide. This is the positive test for the presence of monosaccharide that has been confirmed by the use of Barfoed's test. All experiments are very simple, only you have to write the principle, equation, materials, procedure, and the results. What are the results? Appearance of brick red PPT at the bottom within 3 to 5 minutes indicates the monosaccharides. But for the disaccharides, precipitate will appear in 6 minutes. But for the disaccharide, precipitate will appear in 6 minutes. We have to count the minute time during the performance of this test. This way we can distinguish whether the given sample solution is monosaccharide or disaccharide. This is the recipe of Barfoed's reagent. So the next experiment is Seliwanoff test for the identification of keto group. Whether the given sugar to us is containing the keto group or it is containing the aldehyde group. Keto group, example of keto group is, ribose sugar, and example of aldehyde containing carbohydrate monosaccharide is glucose. Glucose is containing aldehyde group. So the reaction, how reaction is proceeded, ketohexoses are first converted into methyl hydroxyfurfural. That upon its reaction with the resorcinol gives the condensation product. So how to perform this experiment, and let's see its color. What is the procedure? Take 2 ml of Seliwanoff reagent in the test tube and 2 to 3 ml of sample solution in it. Heat the mixture gently. We have to heat the reaction mixture gently. What will happen? Formation of red color shows the presence of keto groups. If the red color will appear, it means the Seliwanoff test is positive, and Seliwanoff test is positive mean the carbohydrate is containing the keto group. The monosaccharide is containing the keto group. It means this. So precaution, avoid spilling of reaction because it is highly corrosive. The reaction is highly corrosive, so avoid spilling of the reaction. Next, reagent. How to make the Seliwanoff reagent? The recipe is given here. You can make this recipe, but this reagent will be given to you ready-made. You will be simply using this reagent to make the reaction. And the significance is given. This test can be used to find the presence of non-glucose sugars in the samples. Okay. Next is the Biles test for the identification of pentoses. Some sugars are pentoses and some sugars are hexoses. In order to find whether sugar is pentose or hexose, we perform the Biles test. For example, this is sugar here. When it is heated, it is converted into The furfural that upon its reaction with orsinol in the presence of FeCl3 is converted into blue-green complex. Let's see how this reaction is performed. Take 2 ml of sample in a test tube and also add 2 ml of Biles reagent. Heat the solution gently. Formation of blue color will indicate the presence of pentoses. Blue color is the indication that the Biles test is positive and the sugar given is the pentose. This is the recipe for the formation of reagents. Okay, this is the last experiment that I'm teaching you about the carbohydrates. I have selected some experiment to teach you. I'm not teaching you all the experiments because you have to write the 22 experiments from the carbohydrate section. All experiments are written very simple way in the experimental manual, and you can see, but some of the experiments that I have selected today to teach you. If you need further guideline, I can further guide you. Okay, so what's the principle? That glucose upon its reaction with 3,5-dinitrosalicylic acid is converted into gluconic acid and 3-amino-5-nitrosalicylic acid. This reaction is performed by the glucose, and this product is then spectrophotometrically quantitated. As much this product is formed, it means glucose is present in high quantity. If this is formed in low quantity, then the glucose is formed in low quantity, and the concentration of this product is correspond to the concentration of the glucose. So what's the strategy to perform this experiment? We need materials: the glucose, dinitrosalicylic acid, test tube, spectrophotometer, and the water bath. Weigh 0.1, make weighing of 0.1 gram of glucose and dissolve it to make the 100 ml. 0.1 ml and dissolve it to make the 100 ml of the solution. Take 100, 200, 300, 400 and 600 microliter of glucose solution in separate test tubes and make the volume up to 10 ml. Take 1 ml from these stock solution in separate test tubes and add 1 ml phosphate buffer. 1 ml phosphate buffer is added, and then add 3 ml of DNS reagent, 1 ml of glucose solution, 1 ml of phosphate buffer, and 3 ml of DNS solution is added. And then it is boiled at water bath in a water bath for 5 minutes. And after the boiling, we take the absorbance at 550 nanometers. So this is the scheme that is followed, that has been shown in the form of table. First of all, we will take the standard glucose solution. It's 100 microliter, 200, 300, 400 and 600. And we add that much ml of water. And total volume become 10 ml. And the final concentration become 0 microgram per ml, 10 microgram, 20 microgram, 30, 40 and 60 microgram per ml. Then we add the buffer, phosphate buffer 1 ml. We will give you ready-made this buffer. And then we add the standard solution of 1 ml. Then we add the sample solution 1 ml. Sample solution mean the sample that is containing glucose of unknown concentration. We don't know the concentration of glucose in the sample, that is called the sample solution. And standard solution, that for which we know the concentration of each sample, that is called the standards. And then we add 3 ml of DNS reagent in all the standard and the sample tube. Then we take the absorbance at 550 nanometer. These are the absorbance that has been taken against the standard solution of these concentration. These are the absorbance, once again I write, these are the absorbance that we took against the standard solution of glucose of these concentration. Against these two set of data, we draw the standard curve. By using this concentration and by using this absorbance, we draw the standard curve. On the x-axis, we draw the glucose concentration, and on the y-axis, we draw the absorbance. And we get a straight line. This kind of straight line we obtain. This straight line is containing R square value, and this standard is line is containing this equation of linear line, of straight line. This straight line equation is containing y, x and y value. This standard line, this equation is containing the value of y and the x. X is the glucose concentration and y is the absorbance. Y is the absorbance and x is the glucose concentration. Okay, I'll move all this thing. By the use of this equation, we know the absorbance of the unknown solution. We know the absorbance of the unknown solution, but we don't know the concentration of the unknown solution. By the use of this equation, we put the absorbance of unknown solution into this equation. This equation is y is equal to 0.0187x plus 0.0886, and the value of y, the absorbance, is 0.675. After putting the value of y in this equation, we solve and we find the concentration of glucose in the unknown solution is 31.37 microgram per ml. The unit of glucose concentration in the unknown sample will be the same as the concentration unit of glucose into the unknown solution. We will also study this experiment from the experimental biochemistry and biotechnology series 1. Okay, so what is the first what experiment we will study today? The grease spot test will be studied, and then the identification of free fatty acid, and then the emulsification of lipid by using the bile salt, and then we will see the sponification value of the corn oil, and then we will see the extraction of total lipids from the brain of the tissue. We will also determine the iodine number. Please mute your mics, all students, and then we will find the concentration of the cholesterol in a sample, in the unknown sample. So let's start one by one. Just like we started the first experiment of the carbohydrate from the identification of the carbohydrates, in the same way we will start the experiments of lipids from the identification of the lipids. For example, if we have the lipid solution, one lipid solution, and one we have the carbohydrate solution, and we have third the protein solution, how will we distinguish between those? So let's see the identification. There are two tests for the identification. One is the grease spot test. Grease spot test is for the identification of the lipids. We will take one filter paper here, just like here. We will write on one side oil, and on one side we will write the water. We will drop a drop. So we will write oil and we will write water on the filter paper. This is the filter paper actually, and we will drop a drop of oil on one side, and we will drop a drop of water on other side, and we will let it dry for 10 minutes, and we will see the translucency, the semi-transparent portion appears where the drop of oil was dropped and the water has been dried out. It is same, this area is same as just like the filter paper. So this kind of grease kind of spot, we will add oil to any cloth or paper. It gives a greasy look. This kind of grease spot test is utilized for the identification of lipids, because we know the lipids are soluble in organic solvents. If we drop a few drops of ether on the oil spot, the oil will be washed away and the same opaque color of filter paper will reappear after the ether treatment. This confirms that the oil lipids give the grease spot test. This is the first identification test for the lipids. Let's move for the second test. Okay, so another one is actually the solubility test. The carbohydrates usually, and the amino acids are solubilized in water, but the oil is not solubilized in water. Sometimes some drops float on the surface of the water, and sometimes some oil drops are set at the bottom of the water. This is the solubility test, second test, that I'm not showing you here, but it is written in the experimental manual. You can read there. Okay, the next experiment number 6.3, we will be talking about the identification of free fatty acid in corn oil, whether the corn oil is containing free fatty acids or not. How to perform this test? For example, this is the free fatty acid, this is the long chain, and this is the carboxyl group. We know this carboxyl group can interact with sodium hydroxide. to make this kind of salt and this water molecule. We need the corn oil, potassium hydroxide, phenolphthalein, and the beaker test tube and the test tube stand to perform this experiment. What are different steps to perform this experiment? This is the procedure. 2 drops of phenolphthalein solution in a test tube, and then pour 2 drops of very dilute alkali solution, and then take 1 gram of oil, dissolve it to make 50 ml of solution, and mix the alkali solution with the oil solution. Mix the alkali solution with the oil solution. This is alkali solution, this is solution of oil. Wait for 2 to 3 minutes. Pink color will disappear as the alkali is neutralized by the fatty acid. Initially when we add the phenolphthalein in NaOH, pink color will appear, but when the NaOH will react with this free fatty acid, NaOH will vanish and the pink color of phenolphthalein will disappear. It means that the corn oil had the free fatty acid that reacted with NaOH, and the pink color of phenolphthalein in basic media is vanished to the colorless. This is the identification test of the free fatty acid in the corn oil. This is the significance of this experiment. You can read the significance. You can check this exercise here. Next is the emulsification test of the lipid. This is very interesting test. Inshallah, we will perform this test. So you already know the lipids. The lipids are not dissolved in water. You already know lipids are not dissolved in water. Okay, but when we eat lipid, they go through our mouth to our stomach. Even in stomach, only the proteins are digested and proteins are broken down, and the carbohydrates, some of them are broken down in the mouth and some are broken down in the small intestine, and the proteins are broken down into the stomach under the low pH, and the lipids are dissolved because of the bile salt. When the bile salt enters in the duodenum, then the lipids are dissolved. The bile salts are containing both the hydrophobic ends and the hydrophilic ends. Who? The bile salt. Okay. So if we add the bile salt to the lipid, they will be dissolved. We can either buy the bile salt or we can get the gallbladder of a chicken. Gallbladder of a chicken is also containing the bile salts. We will get the gallbladder from a shop of chicken, and we will get the dark green color of its salts from the gallbladder, and we will mix that solution. We will mix these all dark green color to make a solution like 5 ml. Okay, how will we perform the experiment then? 2 ml of water is taken, and 2 ml of dilute bile acid is added. Then 2 drops of corn oil is added, and we shake the vigorously. We take 2 tubes. Very bad. 2 tubes we take. In 1 tube, we add 2 ml bile salt, and in 1 tube we add 2 ml water. 2 ml bile salt. Okay. And then we add 2 ml of corn oil in each. 2 ml of corn oil in each. CORN, corn oil. Corn oil here and 2 ml of corn oil here. But the corn oil will only be solubilized where the bile salt is added. When the bile salt is added. But the oil in the second tube will be appeared in the form of suspension or insoluble form. This way we can know that the bile salts can dissolve, emulsify the lipids. Next test is the spanification of the corn oil. We already know that triglycerides are present in the oils and fats. We know already. And during the soap formation, these triglycerides react with the potassium hydroxide to make the soap and the glycerol. How we know the quantity of KOH that is utilized to make the soap to sponify the 1 gram of the fat into the soap? We will know through this experiment. Dissolve 1 gram of corn oil in 3 ml of ethanol. हम स्पोनिफिकेशन वैल्यू मालूम करने जा रहे थे, ठीक है। हमने क्या किया, 1 ग्राम ऑफ कॉर्न ऑयल लिया और 3 ml एथेनॉल में डिसॉल्व किया। फिर हमने क्या किया, उसको राउंड बॉटम फ्लास्क में ट्रांसफर कर दिया सारे मटेरियल को, और 7 ml और एथेनॉल ऐड की। यानी कि 1 ml कॉर्न ऑयल लेके उसका 10 ml हमने बना लिया। 1 ml कॉर्न ऑयल लेके उसका हमने 10 ml बना लिया। 3 ml पहले डाला और 7 ml बाद में डाला, ठीक है। फिर हमने क्या किया, 25 ml हमने क्या लिया, अल्कोहलिक KOH लिया, पोटेशियम हाइड्रोक्साइड का सोल्यूशन लिया 0.5 नॉर्मल, that is made in the alcohol, that is called the alcoholic KOH. Then we set up the blank containing all the region present except the oil, ठीक है। तो हमने क्या करना है जी, हमने ऑयल की जगह जो है ना वाटर ऐड करना है। एक ट्यूब में हमने क्या किया जी, ऑयल है जी, ऑयल ऑयल ऑयल, एक में ऑयल है और एक ट्यूब में क्या है जी, सिर्फ वाटर है, ठीक है, DW डिस्टिल्ड वाटर है, ठीक है। फिर हमने क्या किया, हमने 25 ml KOH डाल दिया, एक KOH इधर भी डाल दिया, KOH KOH, ठीक है, हमने इधर भी KOH डाल दिया, एंड वी आल्सो एडेड KOH here, somewhere here, we added KOH here, we added KOH here. Okay, we mixed and we waited, incubate the both flasks in the boiling water for 30 minutes. And then we added phenolphthalein solution and we titrate it against 0.5 normal HCl, and we titrated it against 0.5 normal HCl. And the difference of the HCl solution used for the titration is utilized to know how much KOH is utilized. Okay, we will see volume of KOH consumed by 1 gram of fat is that the volume of, you already know, the KOH added will be utilized to make the soap, and the extra KOH will be utilized to make its titration with HCl, and extra KOH will be utilized to make reaction with KOH. So the extra KOH will be determined to find how much KOH is utilized actually to sponify this triglyceride. We will see in the next slide. Okay, this is the blank and this is the test. For example, blank is utilizing 20 ml of HCl and test is utilizing 10 ml of HCl. The difference is 10 ml. For example, 10 ml of volume of KOH consumed by 1 gram of fat is, for example, 10 ml. And the 100 ml of 1 normal KOH contains 56 gram of KOH. This is the molecular weight of KOH. In the same way, how much KOH is present in 10 ml? How much KOH is present in 10 ml? This becomes equal to 0.28 gram of KOH, that is equal to 280 milligram. So the sponification value is 280 milligram. 280 milligram of KOH utilized to sponify the 1 gram. To sponify the 1 gram of fat. That's why sponification value of the oil is 280 milligram. Okay, so how to extract the lipids? Whatever we need to analyze, we have to have the sample. How will we extract the total lipids from the mouse spring? For example, we sacrifice a mice, we rupture open its brain and we have to extract the lipids from its brain. We will take 1 gram of wet brain tissue and homogenize in 5 ml of 0.5% acetic acid. 0.5% acetic acid. Okay, so we will incubate it on ice for 5 minutes and then we will centrifuge it at 300,000 RPM for 5 minutes. RPM mean revolution per minute. Then we will discard the clear supernatant and we will repeat the step 2 and 3. We will repeat the step 2 and 3. Then we will take the supernatant and we will add 40 ml of chloroform methanol solution. And we will again mix it. We will mince it, mean just like we will grind it. and we will allow to stand it for 5 minutes. We will homogenize and centrifuge again at 3000 rpm for 5 minutes. We will evaporate. We will take the supernatant. We will take the supernatant and we will evaporate the solution. After we evaporate, the extracted lipids are obtained at the bottom. The solvent is evaporated and we obtain the total lipids. So, for example, we obtained the 0.001 gram of lipids, and initially we utilized the 1 gram of the brain tissue. Initially, we utilized the 1 gram of brain tissue, and the percentage of fat is 0.1%, and the percentage of total lipid in the mouse brain is 0.1%. This way, we can find the percentage of fats in the brain tissue.